Welcome to Tackling Antimicrobial Resistance, a new NIHR podcast series that aims to shine a light on the threat posed by antimicrobial resistance to UK and global healthcare and the role research can play in tackling that threat. I'm William Hope. I'm the Dame Sally Davies Chair of AMR Research at the University of Liverpool and also the co-lead for infection at NIHR. Each week I'll be talking to experts in antimicrobial resistance to get their views on what can and should be done to tackle AMR as the NIHR brings together stakeholders to create a UK-wide approach. Well, hello, everybody. It's my very great pleasure to welcome Professor David Paterson, who is Professor of Medicine at the National University of Singapore and Director of the recently launched Advanced ID. David, welcome. It's very nice to see you.
Thank you so much for having me, William.
So, David, I thought we would just start by having you tell us about your career, how it all started and how you got to where you are now.
Yeah, thanks, William. I'm a clinician at heart, and I became interested in infectious diseases because it was the one specialty that covered all specialties. And, we've obviously patients get infections of their heart or their lungs or their knee or their wherever. And so I liked that sort of breadth of coverage as a clinician. And then I got some very good advice when I was starting to be interested in research, and that was study what you see. So it wouldn't be that applicable for me to study a disease that's only found in, a certain state in Northeast India. But antibiotic resistance is something that all doctors, and particularly infectious diseases doctors, see every day of the week. And that's really what I got interested in as a researcher. And I guess the flow on from that is that as a clinician, I realized that it's one thing being able to find things that you can publish, but what's much more important is things that are actually going to influence clinical practice. And that for some people is, working out what are the optimal doses, whether there's a new antibiotic that might be applicable to certain type of infections. And in my case, the way my research career has evolved is wanting to do studies and particularly clinical trials evaluating different antibiotic strategies. So that's been, I guess, the course of my career in a nutshell, but very much, I guess, patient-focused and wanting to make a big difference to clinical outcomes.
So you trained, and I know because I followed you in Brisbane, and then can you reflect on that time when you went to the United States to train, didn't you? And actually, can you tell us about those decisions and that time?
Oh, absolutely. So, Australia is a great place to practice infectious diseases, and then I went to the United States because I wanted to do research. And I did reflect to one colleague, someone would knock on my door in Australia and say, oh, I've seen the greatest case of leprosy or of whatever infection X. Whereas in an academic medical center in the United States, someone would knock on my door and say, oh, I just got a paper accepted in the New England Journal or JAMA or Lancet. And that was a noticeable difference at that time, that this was in the mid-1990s, that certainly from an Australian perspective, there seemed to be more research opportunities overseas. And I certainly did enjoy the research environment in the United States. And I guess one of the things of being a scientist or a doctor is that you can work in many parts of the world. And then my career took me to southern Italy for 2 years, which was quite sort of a detour, working in Sicily, but that was fascinating. Then back to the US, back to Australia, and now to Singapore. where I sort of interact on a daily basis with clinicians in many, many different parts of Asia. So I think certainly for young doctors, I know in the United States they have difficulty attracting trainees into infectious diseases. I think that's mainly for financial reasons. But infectious diseases and microbiology and science are great careers because of the huge range of opportunities they give to us. And I've certainly felt that I've exploited those in being able to travel in different parts of the world and work in different parts of the world.
Can I, David, take you back to the heady days of Brisbane? And do you remember there was an old infectious diseases ward, Wattlebury, and a very famous unit, and we both learned in that unit. And on Tuesdays, we'd have to go up to the surgical ward 4F and go through notes that were about 3 feet deep, summarizing all of the microbiology. And there was extensive drug resistance back then, extensive. So, and my question is, that was just part of the job. We didn't think, or I didn't think, that those patients were particularly interesting. This was in the height of the HIV/AIDS epidemic, which was, in inverted commas, the interesting parts of infection. What's changed about that?
Yes. So even though, we had many orthopedic patients with MRSA infections, we still had vancomycin. We also started in our intensive care unit and burns unit starting to see patients with ESBL producing organisms, but we still had meropenem. And at that time with HIV, we had patients who unfortunately progressed to AIDS, and we had many, many, many deaths. And through that journey, opportunistic infections and all manner of drug interactions and unforeseen things. And someone who is very famous in the world of infectious diseases said to me, "Why are you studying antibiotic resistance? That's something for drug companies to be interested in, and they can sort of tinker around the edges." But it really was not foreseen at that stage that we would be in the situation where we are at the moment, where we've gone from, yes, we had antibiotic resistance, but we always had options. Whereas now, when I talk to my colleagues in some different parts of Asia, they have infections for which they have no antibiotics at all. And part of that is access. There might be antibiotics in some parts of the world which could treat these infections, but they're not available in those parts of the world. And there are also situations, though, where they truly are in a corner where they have untreatable bacterial infections. And, that's clearly a situation where none of us want to be in. Looking back, I've had patients who've said to me, "I'm only alive today because of penicillin." These were people who were children in the 1940s, and they were the early patients who received penicillin, and their older siblings may have even died from lack of antibiotics because they were in the pre-penicillin era. And so it is sort of confronting to feel that we're now, in some situations, in that post-antibiotic era where we don't have options to treat patients. And so that's something that really needs urgent attention from the research point of view, and also, obviously, many different aspects of how we're going to conquer that difficult issue.
Why, in retrospect, David, wasn't this problem foreseen? I mean, it was sitting there or hiding in plain sight, as the saying goes. Why didn't we foresee this?
Yeah, I think it was I guess part of the issue was the global financial crisis. Maybe that was about 15 years ago or so. And many companies then pulled out of antibiotic discovery and development. And yet at the same time, the germs kept on evolving. They kept on transmitting antibiotic resistance genes between each other. We saw an enormous increase in use of antibiotics in agriculture. And so we had sort of a confluence of things that were promoting antibiotic resistance. Previously, we'd always been bailed out. When a new antibiotic resistance mechanism became available, there was a new antibiotic or a raft of new antibiotics to help with that situation. But for the first time, and this is really VRE and then carbapenem-resistant gram-negatives, we had situations where we didn't have new antibiotics available. Now, that situation was resolved to a large degree with VRE and MRSA, of course, but with the more resistant gram-negative bacteria, we didn't have new antibiotics available. And that's an ongoing situation. So we still haven't caught up. We still haven't worked out ways in which the pharmaceutical industry can really have a sustainable antibiotic discovery and development program. And I think that is really a global issue. There are a lot of thoughts as to how to solve that problem, but I think it's an ongoing problem. It's not been resolved yet.
Okay, so wind the clock forwards now to when you actually had gone back to Brisbane or come back to Brisbane from the United States. You led the Merino study, which had far-reaching consequences for infection of multi-drug resistant pathogens. Do you want to just tell us a little about what that study is? And especially, David, seeing that some of us or people listening to this may not know what ESBLs are, maybe just explain sort of carefully what those terms mean and why Merino was important.
Yeah. So I'll go back on a little bit of a history lesson. In 1964, an antibiotic called ampicillin was commercially released. And it only took until 1965 that they found ampicillin resistance that was due to a clever, I was going to say a clever idea of a bacteria. Obviously, bacteria don't have ideas, but a mechanism by which the bacteria would actually produce an enzyme that would destroy the ampicillin and make it inactive. And that enzyme is, I think we call a beta-lactamase. And at that time, it was found that the gene that encoded the beta-lactamase was actually on a thing called a plasmid, which could spread from bacteria to bacteria. And obviously that multiplies the effect of resistance. Then if we fast forward to the mid-1980s, a type of antibiotic, which we call the third generation cephalosporins, were introduced into Europe. And subsequently, these became the world's leading sellers in terms of injectable antibiotics, things like ceftriaxone. And it was a very similar story. Within a year of the commercial release of the third generation cephalosporins, bacteria were found that were resistant to them. And the mechanism of resistance was again the production of a beta-lactamase, an enzyme that would actually hydrolyze, it would destroy part of the antibiotic. Now, these were not the simple beta-lactamases that destroyed ampicillin. These were, I guess, derivatives of some earlier beta-lactamases, and so they were called extended spectrum beta-lactamases. They had a more extended spectrum than I guess the parents from which they were derived. And we call these extended spectrum beta-lactamases ESBLs. Now, they typically ESBL-producing bacteria were resistant to many, many different antibiotics, and we didn't have many antibiotics left to treat them. And the idea of the Merino trial was to work out whether one particular antibiotic called piperacillin tazobactam, which actually took over the mantle of the world's biggest selling injectable antibiotic from ceftriaxone, the third generation cephalosporin, was that antibiotic good enough for serious infections with ESBL-producing organisms? We always had a class of antibiotics, the carbapenems, which you've heard me mention. One example of that is meropenem. They are intrinsically not destroyed by the ESBL, so they've always had a good track record. But could another antibiotic, piperacillin tazobactam, compete? Now, both of these antibiotics were generic, and still are generic, so no drug companies were interested in this trial. And so it's what we call an investigator-initiated or a clinician-initiated trial. And one thing about trials that doctors organise is that, we don't have 50, 70, 100 million pounds to spend on the trial. We typically have less than 5 million pounds. And so, sure, there are what might be regarded as weaknesses or holes in the design of these sort of pragmatic trials, which are done really with the purpose of informing clinical decisions. They're not for getting a drug approved by the FDA or EMA or whoever. So the reason we called it the Merino trial is MER stands for meropenem. And the trial was being done in Australia and New Zealand, where we have a lot of sheep. But then we worked out that, we don't actually have enough patients with these ESBL-producing organisms in Australia and New Zealand. And so we then moved to Singapore, where they don't have many sheep, I can assure you of that. But we kept the name, the Merino trial. We ended up doing it in Turkey and Canada and South Africa, Italy. So it had sort of global reach. And what we anticipated finding was that piperacillin tazobactam would be just as good as meropenem. And that would have had a lot of advantages because one, I guess, truism about antibiotic use is the more you use it, the quicker you lose it. So if we use meropenem, a carbapenem, all of the time, we know that that's going to ramp up resistance to the carbapenems. And we know that after the carbapenems, there are not many options. And so we really were hoping that piperacillin tazobactam would be just as good as meropenem. Unfortunately, our hypothesis was wrong, and meropenem patients treated with meropenem were less likely to die than people who received piperacillin tazobactam. So that was obviously a very informative result in terms of clinical practice. It was not the sort of result that our antibiotic stewardship teams wanted to hear. So antibiotic stewardship is this concept of in hospitals, not everyone is really up to speed about what is the best antibiotic to use for any given situation. And antibiotic stewardship is really finding the best antibiotic for certain situations. And that's ensuring that there's a diversity of antibiotics used rather than doctors just focusing on one or two antibiotics. So I'm sure most antibiotic stewardship teams didn't want to have this emphasis on meropenem, but that was the fact, that we found that meropenem was associated with a better outcome for our patients. And so that trial, I think, had well over 200,000 page views on JAMA. It was voted by doctors at the Infectious Disease Society of America meeting the year it was published, 2018, to be the trial that year that was most likely to change people's practice. So that's, I guess, why I wanted to get into clinical trials regarding antibiotics, because I did want to make an impact. So that was an interesting experience, and all of us who are involved in the trial learned a lot about doing trials, and doing trials that doctors want rather than trials that are just there to get regulatory approval. And I think there are differences in how you approach these sort of clinician-initiated or investigator-initiated trials compared to how we behave in what I might call regulatory trials.
Okay, so Merino was real world. It was elegant. It was simple. It took a lifetime of training to design and deliver, right? So it's it's popular amongst the community because for the first time there's some evidence rather than just personal opinion. And can you reflect, David, what is the difficulty about enrolling multi-drug resistant or antibiotic resistant pathogens into clinical studies? Why is Merino so unique? If drug resistance is so common, why was your study so unique?
Yeah, I think it was because we took this sort of pragmatic approach. So we know when various companies have come to study, for example, carbapenem-resistant bacteria, so the next level up from ESBL producers, they have had a lot of trouble recruiting into these studies and have recruited small numbers at a very, very high cost. And there are all sorts of figures flying around, whether or not they're entirely accurate, of, costing $1 million to recruit one patient. Part of the difficulty is that many of these regulatory trials have got a lot of exclusion criteria. So typically when you're developing a new drug, for example, a new antibiotic, you want to test it in a pretty clean population. You don't want to have people who are on dialysis. You don't want to have people who've got cirrhosis. You sort of avoid the really sick end of the spectrum. And yet that is exactly the spectrum in which we see carbapenem-resistant organisms. We typically find these patients in intensive care units or in hematology units. Often they've got a lot of other medical problems. And so typically these patients were excluded from the trials. And that's, I guess, a reason why we need to approach trials in this particular area from a much more pragmatic viewpoint, meaning we should be willing to enroll people even if they're very sick, because in fact these are the people who really do end up with these organisms. So to make these trials as generalizable as possible. The other thing is that I remember hearing about this bank robber called Sutton, and there's a sort of an adage in infectious diseases called Sutton's Law, and that is you should go for where the money is. And that's exactly what they said to Sutton. Well, why do you rob banks? And he said, that's where the money is. And so in infectious diseases, if someone's got a cough and they've got this fever you can't work out, will go into the chest. And it's the same, I guess, adage in trials of very resistant bacteria. They're absolutely widespread in Asia, in Latin America, in parts of Africa, and yet, sure, they do occur in Western Europe, in the United States, in Canada, and in Australia, but we have such a big attention to infection control that the numbers are several orders of magnitude lower than exist in Asia. And so in a way, that's one of the big reasons why I've moved to Singapore, so I can do these trials on these resistant organisms where the action is, rather than trying to recruit the one every couple of months very tragic patient that we'd have in Australia, whereas these types of organisms are seen very much on a daily basis in many hospitals in Asia.
So, yes, and part of the introduction, we alluded to Advance ID. So why don't you tell us about Advance ID now?
Yeah. So this network, and Advance ID stands for Advancing Clinical Evidence in Infectious Diseases, which is very much my sort of adage. We've been set up by funding from the Wellcome Trust and also a number of institutions in Singapore to establish a network across Asia. And at the moment, we've got more than 35 hospitals from India, Pakistan, Bangladesh, Thailand, Vietnam, Malaysia, Indonesia. And I guess what you might call our peacetime activity is to study AMR, antimicrobial resistance, and particularly to do clinical trials of the most resistant organisms here where they exist. And while you may say, well, those trials are targeted at populations in Asia, and do they really represent, can they help if I'm looking after a patient in Liverpool or Leeds or in Brisbane or Calgary? Can those results help me? Well, I think the answer is yes, because here is rigorous randomized trial data showing how best we can treat these particular infections. You might not see them every day if you live in Manchester, but when you do come to have these infections, hopefully the data we generate will be generalizable to you wherever you are in the world if you see this particular pattern of resistance. So that's what's our peacetime activity. But I guess we've also got to remember that we had a lot of trouble, I guess, everywhere in the world except the UK, in getting clinical trials set up for COVID. And if there is a COVID-28, if we've got a network of sites throughout Asia who are very competent doing clinical trials, because that's what they're doing every day, we are hopeful that we could pivot very rapidly and study the next pandemic in very much get things organized very quickly, just as you did in the UK with recovery. And so that, I guess, is the secondary aim, but there's no shortage of people, unfortunately, dying from AMR throughout the world. So I think even in peacetime, we're going to have our work cut out. But we really want to deliver very pragmatic findings that can be used by doctors throughout the world to improve the care of their patients.
So, David, in a minute, I'm going to ask you to explain to everyone what a platform study is. But just before I do that, what types of infections are we talking about? You very nicely gave the history of ESBLs, and they're sort of almost ordinary these days. What are we talking about for Advance ID?
Yeah. So really the next level up from ESBLs are these carbapenem-resistant organisms. So we use carbapenems to treat ESBL producers, but now that is lost. And so we've got a variety of bacteria that are carbapenem resistant. And the go-to drug has been a drug that was approved in the 1950s called colistin. And by, I think, the early 1970s, people stopped using it because it was so toxic. And all these new antibiotics became available, and it was just left on the shelf. But then now that we have these carbapenem-resistant organisms, colistin was brought back because the germs had forgotten about it, and it was typically very active against a very broad range, including carbapenem-resistant organisms. But two things. First of all, as you'd anticipate, there has been development of colistin resistance. And secondly, I don't think it's fair that in lower and middle income countries, they're still having to use a very, very toxic antibiotic when in other parts of the world there may be available new antibiotics which are pretty much clean in terms of their toxicity profiles. So our first studies are going to be looking at these carbapenem-resistant organisms and comparing a variety of antibiotics, but also combinations of antibiotics to see if we can fool these very resistant organisms by approaching the problem in two different ways. Now, we know in HIV or in TB, people don't get a single drug. They get multiple drugs. They get combinations of drugs. We've not frequently done that in terms of antibiotics because there's a fear that all it does is just multiply toxicity rather than multiply efficacy. But in the test tube and in some sophisticated, models of infection, combinations of antibiotics do kill better than a single antibiotic given by itself. So, but again, that's not the sort of thing the pharmaceutical industry is typically interested in studying. They want to get their drug approved. And while clinicians may be toying with using combinations of antibiotics, they've never been proven to improve patient outcomes. And that's what we would like to do.
And you're proposing to do this via generation, you use the word networks, but of platforms. And so you're building on some of the ideas that were not pioneered, but made extensive use of in the pandemic. And you alluded to recovery, and there are others in the UK, REMAP-CAP, which was a study led from Australia. So what is a platform? Just explain that.
Yeah. So the normal way we do trials is you compare drug A versus drug B. You get everything set up for that trial. You compare drug A and drug B. You finish the trial, you close the shop, and that's the trial done and dusted. But that takes a lot of effort to that startup of getting all the different hospitals together. That whole process is very labor intensive. And particularly, when we're not talking about having ability to have, £100 million just for one study, a much more economical way of doing things is to start broad and have a number of different related trials going on simultaneously. So I've talked about gram-negative bacteria, and there are a number of different gram-negative bacteria. There's obviously E. coli that people have heard about, or Klebsiella. There's another one called Pseudomonas or Acinetobacter, and each of them have their own subtleties. But it makes sense to have trials of these bacteria going on simultaneously. And the other advantage of this, setting up this platform, is if we find that one particular drug or combination turns out to be a dud, it's just ineffective or it's toxic, and at the same time, a company may have a new antibiotic, or a research group may have some new data saying that a certain combination is particularly effective in their models. With a platform, instead of having to go back to the beginning and start this whole development process, we could insert that new drug or that new combination into the platform and try and make things as seamless as possible. So I guess the idea is to try and be as economical and as effective as possible in getting many answers in the shortest period of time and with the least expense. So it is a big organizational task, but it's being done at the moment with MRSA, and it's been done with COVID. So there certainly is, every possibility that we can do it with these gram-negative bacteria.
A concept that I've always known, but is more obvious, of course, is that often the evidence that we generate is quickly out of date because evolution and the emergence of resistance is dynamic. And so there needs to be continuous knowledge generation. And that's why infectious diseases is different from other areas like cancer. Maybe you could just reflect on that. So David Livermore, who we both know well, refers to these as black swan events, for example. There are always new events occurring. So
to you and me, William, a black swan is an everyday event in Australia.
We better explain that, but yeah, okay.
That's right. But the concept of these platform trials is that they could be eternal, meaning that if there are ongoing problems to address, once you find, for example, that drug A or combination A appears to be the best, it wouldn't be surprising if new drugs became available or new subtleties in terms of the antibiotic resistance profile of a bacteria became available or occurred. And so then there's the opportunity of really, well, is drug A still the best in this new scenario or up against this new antibiotic? So instead of having to start the whole thing again, the concept of the platform is that it could just keep going and going and going. And we know that the problems are never going to go away. Or if they go away, fantastic, we can close up shop. But certainly in my career, antibiotic resistance is a thing that's not going away. And so people have made these models and these predictions that by 2050, more people will die from infections due to AMR than will die from cancer. So it might be that the people with the cancer platforms are shutting up shop. But I suspect that the students that I'm training at the moment, I'm hoping that they will take over from me running the platform. Hope because I want to invest in the next generation of researchers. I guess wish that the problem would go away, but I don't think it is going to go away, the problem of AMR.
So, David, it's curious then, this pivot. I mean, obviously you have to go to the places where there are the organisms and cases to study. That seems blindingly obvious. So then just tell me, what is the role of countries like the United Kingdom, continental European countries, United States? Should they have platforms? What's their role in tackling this global problem of drug resistance? What role do they play?
So I think the reality is that discovery and development is very much occurring in high-income settings. I think it's a reality that we're not going to see in Indonesia or Bangladesh, development of new antibiotics. But I think one big role in high-income settings is the discovery of new chemicals, but also their evaluation in very sophisticated ways to determine whether they are good options. And of course, this problem of resistance, the UK, for example, is certainly not immune to that. It's not seen perhaps to the same huge population level that it's seen in Asia. But I'm sure it gets on the front page of papers if there's a substantial outbreak in a particular hospital, and it's affecting a lot of people. So I think to sort of put that into a summary, science and highly sophisticated development of concepts of dosing of antibiotics, of discovery of antibiotics, of determination of mechanisms of antibiotic resistance are going to go on in the UK. And there's been obviously a hugely strong heritage of that. And I think it also makes sense for patients in the UK to be part of these platforms. They might not be enrolled to the same level as elsewhere in the world. But one of the beauties of a platform is that you can also think of important clinical questions. People with garden variety E. coli that might not be particularly resistant to antibiotics, it's probably one of the least studied infections in the world in terms of clinical trials. How long should we treat it for? Is there any benefit of 2 antibiotics? How can we prevent a person who's got an E. coli infection from getting a second one? And so there's many, many questions that could be answered really by cooperation. My sort of imagination of this platform is that it's covering a variety of different bacteria with a variety of different levels of resistance. It might be heavily recruiting in one particular area in Asia. There might be overweight of recruiting for other particular components of this platform in the UK. But I really do feel it's most likely to be effective if it is a global cooperative platform. And people in Canada or the US, UK, Western Europe, Australia, Asia, all cooperating on the same research program, I think that's going to have a huge, huge impact for patients around the world. And that's really what I'd like to work together with others on developing.
So it's interesting, isn't it? That doesn't quite chime with the world's politics just at the moment, which is
exactly.
maybe more inward looking. So one of the great problems of our time requires a global view and global solutions at a time when we seem less inclined to think that way. So maybe we can just drive that point home about why someone in the United Kingdom would have any concerns or care about what happened to someone with a multi-drug resistant organism in Singapore. Just tell us why someone in the UK should care about that.
So, all of us like to travel. That's certainly one thing. And I've had no shortage of patients who, I've gone to Thailand or to Bali. You can hire a motorbike for, £10 a day. I might have a crack, end up having an accident, end up in hospital, get repatriated back home. The germ that you have, which is a multi-resistant germ that you acquired in Indonesia or Thailand, is unfortunately transmissible, not through breathing, but through contact typically, or through equipment. And, soon enough, we could have an outbreak of a very resistant organism in any part of the world, whether that's in Wales or Scotland or Northern Ireland or in, Western Australia. It's the same concept. What we do unfortunately know is that sometimes these outbreaks happen under the radar. And that's a bad situation because we can end up with these germs getting stuck in a particular ICU or unit of a hospital. We call it, they get endemic, meaning that they've been introduced and then they've spread without us realizing. And now we're in a big problem. It's a problem that we can't eradicate and we have to live with. And that's really concerning when it's often in people who are at their most vulnerable. It might be a cancer unit, it might be a geriatric unit. And when you combine a germ that is difficult to treat with a person who's got a lot of other medical issues and might be frail, they might be immunosuppressed, that combination, unfortunately, can sometimes have fatal consequences. And that's something we really want to avoid, the situation where a person dies from an infection and doesn't get the chance to, to fight their leukemia or move back home from hospital. So unfortunately, antimicrobial resistance is a global issue, and everywhere in the world, our hospitals and our nursing homes and our communities are facing it, certainly to different levels in different parts of the world, but it's not something we can ignore.
So, David, that seems like the perfect note to end on, a call to arms and a reason that we should all be interested in care. Thank you, David, for your very erudite and simple explanations for this very complex problem. Thank you for the contributions that you've made and continue to make. We wish Advance ID all the very best and recognize that it's going to provide solutions that are important for all of us. And thank you very much for your time from Singapore today.
No worries.
Today, David. And keep the good work up. Thank you. Bye-bye.
Thanks, William. Bye-bye then.
Thank you for listening to this episode of Tackling Antimicrobial Resistance, a short NIHR podcast series hosted by me, Professor William Hope. If you have any thoughts or comments on this or any other episode, please contact us via our Twitter channel at NIHR Community. And for more information about the NIHR, you can visit our website, nihr.ac.uk.